Okay, so metaphase 1, I'm going to put my centrioles back to the poles. It says chromosome pairs line up at the equator, spindle fibers attached to the centromeres. So I'm going to take my homologous pairs and I am going to put them at the equator. They're going to stay lined up, they're going to stay matched up, and I am still going to represent my crossing over. Blue, that should be blue. And let's see, this guy goes here, this guy goes here. So my homologous pairs, and again, and don't forget crossing over could happen one place, two places, three places, four places. It's random every time. And last but not least, our spindle fibers attach to our centromeres. This should all be kind of familiar to you guys. And that's metaphase 1. Okay? Let's move to anaphase 1. This is easier. Friday I did this on the board and I can never remember what colors were where. Okay, so anaphase 1. Again, I am going to draw my centrioles where they're supposed to be. As you can see, the cell is starting to elongate, getting ready to kind of make two different cells. Matching pairs are pulled to opposite poles by contracting spindle fibers. So I'm going to just draw my spindle fibers like this and make them kind of short. And then I am going to attach my chromosomes. I had my light blue one right here. And I had my darker blue one right here. And I had my pink one right here. And I had my red one right here. So don't forget, these guys are duplicated chromosomes, and don't forget to show your crossing over again. Crossing over. Crossing over. And that's it in anaphase 1. Hopefully this looks kind of familiar to you guys. We're going to move down to telophase 1. A few things are going on. You guys should have kind of a little indentation like that on yours. So nuclear envelope may or may not reappear. So let's go ahead and draw that it reappears. Okay, so I'm going to draw my nuclear envelope. I got one here. I got one here. DNA begins to uncoil. So I'm going to draw my chromosomes, my duplicated chromosomes. They're going to look really kind of thin. So I'm going to make my structure. I've got my light blue one right here, and I'm just going to make it thinner, and I am going to show my crossing over still. And I had my darker blue one right here. You can see how my chromosomes are thinner. There we go. And I had my pink one over here. Really kind of uncoiling. My red one right here. And put a little show my crossing over. Show my crossing over. Cleavage furrow appears. So this little indentation right here, this is your cleavage furrow. I'm going to draw a dotted line right across here. to show cytokinesis starting to occur, complete pinching off of the cell. So the cell kind of starts eventually, this edge and this edge of the cell membrane are going to come together, and we're going to end up with two cells, okay? I'm going to put N right here and N right here to remember that these guys are haploid, okay? It's half the number of what we started with. So right now, each of these chromosomes, or each of these cells are going to have 23 duplicated chromosomes, okay? So I'm just going to draw my nucleus. I'm going to draw my full nucleus right here. So I've got two different cells. I'm going to hurry and put my chromosomes in here. Still going to add a little crossing over. Still going to add a little crossing over. My colors here. One more chromosome, crossing over, our centrioles, I don't have it written down here, but our centrioles usually are going to kind of move together somewhere in the cell that isn't necessarily at the opposite poles. So we got two haploid cells, each with 23 chromosomes. So at this point, what we are going to do is we're going to pull these chromatids apart. And that's how we're going to end up with 23 in each sperm or in each egg. So we are going to move to meiosis 2, prophase 2. Centrioles move to opposite poles, so of course we've got more drawing because now we have two cells we have to deal with. Centrioles move to opposite poles. Nuclear envelope breaks down if it reappeared, so I'm going to draw us a dotted line around my nucleus so it looks like it is getting ready to disappear. I'm going to stick my chromosomes back in my cell, but I am going to make them thicker and more condensed. So I've got my light blue guy here. I've got my dark blue guy here. Again, show your crossing over. And in my other cell, I had a pink chromosome, and now he's nice and condensed. Show my crossing over. And I have my red one right here. He's nice and condensed. Okay. So at this point, we're going to move to metaphase 2. Metaphase 2, our chromosomes are going to align themselves at the equator. So I'm going to put my centrioles back over here. My nuclear envelope would have completely disappeared by now. So my chromosomes are free to move to the center. So I'm going to put my light blue one right here in the middle and my dark blue one right here. And I'm going to show my crossing over. I'm going to attach my spindle fibers. Oh, I forgot to draw my spindle fibers reappearing in prophase 2. Okay, then I'm going to put my other chromosomes at the equator. Nice and thick. More crossing over. More crossing over. spindle fibers attach at the centromeres. Hopefully you guys can kind of see where we're going here. Those sister chromatids are about to be pulled apart, okay? Difference between anaphase 1 and anaphase 2 is this. Anaphase 2, the sister chromatids get pulled apart. Anaphase 1, the pairs are being pulled apart, okay? Okay, so anaphase 2, I can't remember what side my colors were on. Okay, so anaphase 2, I'm going to draw my centrioles again. Centrioles. And by the way, these are considered your notes. So on your quiz on Friday, you can use these. Okay, so I'm going to draw my little centrioles very close, or my spindle fibers very close to my centrioles. And this is where I'm going to show that my chromatids get pulled apart, okay? So I'm going to have one chromatid here, one blue, one light blue chromatid here, and I have one dark blue chromatid here. Again, I'm going to show my crossing over. We're very close to being done. And then on my second cell, I am going to show that my chromatids are pulled apart. I got a pink one here, and a pink guy here, and I got a red guy here, and a red guy here. I'm going to show my crossing over, and there we go. And I'm going to add right here, I'm going to say sister chromatids are pulled apart. Sometimes that's an easy way to distinguish between anaphase 2 and anaphase 1. Anaphase 1, homologous pairs are pulled apart. Okay, anaphase 2, sister chromatids are pulled apart. Okay. Our very last, we just buzzed right by. Okay, telophase 1 is our last one. I've got these little boxes, but you guys should have your cleavage furrow. Nuclear envelope reappears. So let's go ahead and draw our nuclear envelope reappearing. Let's do our dotted line for our division of cytoplasm and cytokinesis. And let's go ahead and draw our sister chromatids and make them thinner. So I have my light blue one here. There are these little, they're single chromosomes now. They're not duplicated. And show my crossing over. Show my crossing over. I've got my light blue here, a little bit of pink, a little bit of red. I have my little pink one right here, and a little pink one right here. I got two blue pens that look the same, and one of them is black ink and one of them is blue. So I keep forgetting which is which. Red, red, and then I'm going to put my little bit of blue here, a little bit of blue. cytokinesis happens, we end up with 4 cells. Each of these are N. I'm going to put N to remember that each of these are haploid. N is equal to 23 chromosomes. I'm going to draw my nuclear envelope completely back. My 4 haploid gametes. I'm going to again draw my chromosomes, and we have almost made it to the finish line. And we had some dark blue ones here, a little bit of red. I've got my pink, a little bit of blue, a little bit of blue. Awesome, if you can put them right over there, that would be great. Thank you, Noah. And my last red chromosome, with a little bit of blue and a little bit of blue. And that is telophase 2. Now, just a quick reminder, if it's sperm that's being made, all 4 of those sperm are functional and they work no problem. If it is oogenesis, if it's an egg that's being made, 1 cell is viable and all the other 3 are called polar bodies. Do you guys remember that from last time? We did like a very kind of rough drawing. So in spermatogenesis, all 4 sperm are good to go. They work no problem. In oogenesis, there's such a uneven division of the cells that actually only 1 egg is viable. Okay, so guys make a lot of sperm and women don't make very many eggs. And I think that's it. We buzzed right by. Okay, for my video people, I'm going to put this right here. Make a little. For those of you who are watching the video, I forgot to start my video recording, so make sure you go back and fill in the picture for prophase 1.